Often called the fight-or-flight stress response, the adrenaline pathway has numerous simultaneous effects throughout the body. This pathway is associated with the sympathetic nervous system, which initiates the fight-or-flight response and aids you in responding to dangerous or stressful situations. This response includes an increase in heart rate, helping deliver more blood to areas of your body that need more oxygen, along with an increase in lung capacity.
In the adrenaline signaling pathway, adrenaline, which is the signaling molecule, is a hydrophilic or polar molecule, which also means that its protein receptors are on the surface of the cell in the cell membrane. This is because hydrophilic molecules are not as easily diffused through the cell membrane, as opposed to hydrophobic molecules, due to the phospholipid bilayer composed of hydrophilic heads and hydrophobic tails. The protein receptors used in the adrenaline signaling pathway are G-protein coupled receptors, or GPCRs, which are integral membrane proteins that are used by cells to convert extracellular signals into intracellular responses. They are typically used in the cell to convert responses to hormones, neurotransmitters, vision, and taste signals.
The main advantage of G-protein coupled receptors is that they produce a rapid and short-lived response. This means that the effect of GPCRs is very immediate, which is helpful when it comes to the fight-or-flight response in the sympathetic nervous system. This is because it prepares the body for stressful situations. Along with this, G-protein coupled receptors can detect external signals and transduce the signal into an amplifying enzyme, whose activity elaborates a second messenger, thus initiating a signaling cascade. Signaling cascades play the role of an intracellular transmitter, which transmits biochemical information between the cell membrane and nucleus. This helps increase the speed of the response in the adrenaline signaling pathway. Additionally, due to their involvement in many human diseases, GPCRs are recognized worldwide as valuable drug targets. However, the main disadvantage to GPCRs includes their lack of specific and high-affinity antibodies. This makes them harder to study and makes the immune system more susceptible to certain kinds of infections, since mutations in genes encoding these proteins are more common and can lead to more diseases. When a GPCR binds a ligand, the ligand triggers a conformational change in the 7-transmembrane region of the receptor. This activates the C-terminus, which then recruits a substance that in turn activates the G-protein associated with the GPCR. Activation of the G-protein initiates a series of intracellular reactions that end ultimately in the generation of some effects, such as increased heart rate.
The first step in epinephrine signaling occurs when the hormone binds to an epinephrine receptor on the cell surface. The hormone triggers the receptor to change shape, converting the receptor to its active form. The activated receptor triggers a cascade of events within the cell, beginning with the activation of a G-protein. The G-protein binds to the activated receptor, releases GDP, and takes up a molecule of GTP. After taking up GTP, the G-protein is released from the receptor and splits into two parts. One of the parts is activated and continues the signaling cascade. Soon, the hormone also leaves the receptor, and the receptor reverts to its inactive form. The G-protein activates an enzyme called adenylyl cyclase. When activated, adenylyl cyclase converts a large number of ATP molecules into signaling molecules, called cyclic AMP, or CAMP. Because CAMP carries the message of the first messenger, epinephrine, into the cell, CAMP is referred to as second messenger. In response to an internal timer, the G-protein soon inactivates itself by taking away GTP, and the subunits reassociate. With the G-protein no longer attached, the adenylyl cyclase turns off and then can no longer convert ATP into CAMP. The CAMP molecules that were produced by adenylyl cyclase continue the signaling cascade by binding to a type of enzyme called a protein kinase. This binding triggers protein kinase A to separate into subunits. The activated protein kinase A subunits perform chemical reactions in which they add phosphate groups to another type of enzyme, called phosphorylase kinase, activating it so it can add a phosphate group to another enzyme in the cascade, called glycogen phosphorylase. This enzyme also becomes activated and produces the cellular response to epinephrine. Glycogen phosphorylase breaks down glycogen into glucose molecules. During the process, the enzyme adds a phosphate group to each of the glucose subunits. Another enzyme removes the phosphate groups from the glucose molecules. Without the phosphate groups, glucose molecules can be transported across the plasma membrane of the cell. Once outside of the cell, the glucose enters the bloodstream and is taken up by other cells and used as fuel. This is a key component of the epinephrine-induced fight-or-flight response.
Epinephrine, also known as adrenaline, is a versatile chemical messenger that signals many different cell types throughout the body. In the lungs, epinephrine binds to the receptors on smooth muscle cells wrapped around the bronchioles. This causes the muscles to relax, dilating the bronchioles and allowing more oxygen into the blood. At the sinoatrial node of the heart, epinephrine stimulates pacemaker cells to beat faster. This increases the rate at which other chemical signals, glucose and oxygen, are circulated to the cells that need them. The epinephrine signal transduction pathway can be shut down in several ways. After several seconds to several minutes following the activation of the alpha G protein, the G protein can actually undergo a self-inactivation process in which it takes a water molecule from the cytoplasm and hydrolyzes the GTP back to GDP. This will decrease the affinity of the G protein for adenylate cyclase, which means it will then dissociate from the cyclase. This will shut down the activity of the adenylate cycle, preventing it from producing any more CAMP secondary messengers. Another way by which the signal pathway can be turned off is if the epinephrine bound to the receptor actually dissociates from the binding site. By dissociating from the receptor, it turns off the receptor's ability to further activate G proteins.
In the process of the adrenaline signaling pathway, adrenaline, or epinephrine, serves as the catecholamine signaling molecule of the G protein-coupled receptors. Catecholamines are a type of neurohormone that are very important in aiding the body within the stress responses, such as increasing heart rate and lung capacity. Adrenoblockers are compounds that inhibit the action of adrenaline along with other catecholamines that control autonomic outflow and some functions of the central nervous system at the adrenergic receptors or inhibit their release. Adrenoblockers are exogenous antagonists because they are drugs produced outside of the body and do not originate within living organisms. Adrenoblockers are highly effective pharmaceuticals that are used broadly in treatment of cardiac diseases. Their physiological effects include the dilation of blood vessels, which lowers blood pressure and slows heart rate, resulting in the opposite effect of the adrenaline signaling pathway. This means that adrenoblockers attach to the G protein-coupled receptors without activating it, thus decreasing the activity of adrenaline and slowing the effects of the adrenaline signaling pathway. With that said, the response is altered by being inhibited and therefore decreasing the heart rate and lung capacity.